The Greenland shark navigates the deep, dark Arctic at an average cruising speed of just 0.3 meters per second. With only about 9 sluggish tailbeats per minute, it moves through the cold water carrying an immense biological weight, a lifespan extending well over 4 centuries. Standard biological assumptions suggest that century-long life requires flawless cellular maintenance. This model of strict resistance assumes an organism's cells must actively and successfully repel metabolic waste and oxidative damage to remain functional. But actual histological analysis of the Greenland shark's myocardium reveals a different reality. These panels show tissue packed with extensive interstitial and perivascular fibrosis. The dense blue-stained areas indicate massive structural scarring and accumulated cellular waste. These sharks do not escape the physiological toll of aging. Their extreme lifespan is built on an extraordinary resilience to structural decay. They accumulate severe tissue damage, yet their hearts continue to beat. Shifting the focus from damage prevention to damage survival changes the model of biogerontology. In the Greenland shark, extreme longevity is achieved through a biological system capable of operating under a massive, century-scale physiological burden. To prove this cardiac degradation is the result of centuries of aging, and not just a structural requirement for living under extreme hydrostatic pressure, researchers needed to isolate specific variables. They established a 3-species control matrix. The deep-sea lantern shark shares its habitat and lineage, but lives only a decade, isolating the factor of time. The short-lived turquoise killifish provides standard aging markers, isolating traits specific to elasmobranchs. The objective is precise: determine if the Greenland shark's cardiac degradation is a deep-water adaptation or a true manifestation of centuries of wear and tear. Any histological anomaly appearing exclusively in the Greenland shark within this matrix represents a functional mechanism for sustaining life across centuries. In standard vertebrate aging, the heart typically develops interstitial and perivascular fibrosis. This is the buildup of extracellular collagen, a stiff structural protein, between muscle cells and around blood vessels. It is a marker for maladaptive cardiac remodeling that usually leads to rigidity and heart failure. Looking directly at this Masson's trichrome staining, we see widespread interstitial collagen networks mapped throughout both the compact and spongy layers of the Greenland shark's myocardium. The blue dye highlights a massive extracellular scar response. To test if this collagen deposition is simply a structural requirement to survive high hydrostatic pressure, we check the deep-sea lantern shark control in panel H. There is a total absence of blue interstitial collagen. Even looking into the spongy layer, the tissue is clear. This evidence rules out the environmental pressure hypothesis. The aged killifish control shows tissue without pervasive blue scarring. Chronological aging alone does not necessitate this specific type of massive extracellular matrix restructuring. This massive non-pathological fibrotic response is an age-related reality entirely unique to the Greenland shark. It accumulates immense scar tissue without triggering heart failure, posing a question about how the organ avoids stiffening into arrest. Beyond the extracellular matrix, we must look inside the cardiomyocytes themselves for canonical markers of oxidative stress, like 3-nitrotyrosine and oxidized lipids. This imagery reveals the massive accumulation of lipofuscin, an undegradable, toxic byproduct of cellular wear and tear. Notice the dark Sudan black B staining. The pigment entirely fills the sarcoplasm of the Greenland shark's cardiac cells.
Because Sudan black B is a standard dye, researchers used fluorescence to verify this dark pigment is truly lipofuscin and not a chemical staining artifact. They subjected the tissue to a photo-bleaching test. After 30 minutes of continuous blue light exposure, standard nuclear stains degrade, but lipofuscin is highly photostable. As the blue fades, the characteristic yellow-brown autofluorescence of lipofuscin remains visible, locked inside the cell. Contrast this intracellular hoarding behavior with the aged killifish control. The killifish also accumulates lipofuscin waste, but it stores it outside the cell, dispersed within the trabecular interspaces. The deep-sea lantern shark lacks this pigment entirely. This proves the massive waste buildup is tied to exceptional chronological age, not the deep-sea habitat. The cardiomyocytes of the Greenland shark uniquely tolerate the internal storage of toxic oxidized lipids. In other functioning vertebrates, this level of intracellular pollution would lead to cellular destruction. To observe exactly how these organelles physically manage this extreme, century-long metabolic waste load, researchers utilized electron microscopy. High-resolution data shows the cellular logistics at play. You can clearly see an abundance of degenerating mitochondria, marked here, actively being swallowed and incorporated into dense, electron-dense autophagomes. The scale of these disposal units is staggering. A three-dimensional reconstruction of a single autophagome measured an exceptionally large volume of 24 cubic micrometers, acting as an internal landfill for oxidized waste. In standard mammalian models, this level of oxidative damage, mitochondrial failure, and severe autophagic load acts as a direct trigger for apoptosis, programmed cell death. Yet researchers found a paradox. When they ran a Tunnel assay to search for dead or dying tissue, they found 0 apoptotic or necrotic cells in the myocardium. Despite the heavy fibrosis and lipofuscin toxicity, the tissue was fully viable. The shark's cells operate with a threshold for structural and metabolic decay that exceeds standard vertebrate limits. It preserves tissue viability under conditions that are mathematically lethal to shorter-lived species. At the macroscopic level, we must explain how a severely fibrotic, structurally stiffened heart physically maintains blood flow for 400 years. The cardiovascular mechanics rely on a specific physiological baseline. The shark operates with a sluggish resting heart rate and low blood pressure, estimated at roughly 2.8 kilopascals. This low pressure allows for structural compensation. The shark's ventral aorta contains loosely organized elastic fibers. When the rigid ventricle pushes a pulse of blood, the aorta expands to absorb the force. This high-stretch arterial system acts as a physical buffer. It absorbs the hemodynamic shock that a fibrotic, stiffened myocardium would otherwise send through the circulatory system. Extreme longevity relies on systemic biomechanical workarounds. The external vascular architecture physically compensates for the irreversible degradation occurring inside the cardiac tissue. A functional body powering through the Arctic for over 4 centuries, driven by a heart displaying massive fibrosis, severe oxidative stress, and immense lipofuscin toxicity. This is the biological reality of the Greenland shark. It illustrates the difference between an idealized goal of preventing cellular damage and the evolutionary reality of surviving it. Extreme vertebrate lifespan does not require the eradication of aging biomarkers. It does not require an ageless physiological state. The Greenland shark indicates a shift for the field of biogerontology. True extreme longevity is found in the evolution of profound physiological tolerance.